Assalamu alaikum. This is Rana Talaha Haider by the subject Plant Physiology. Today, my presentation on phytochrome mechanism in plants. About the introduction of phytochrome. Phytochrome is a photoreceptor of pigment that plants and some algae and fungi use to detect the light. So it is sensitive to the light in the red and far red region of the visible spectrum, in which contain the pigment that control growth and flowering in many plants. So it is biochemically, phytochrome is a protein with a billion and billion chromophore present in it. Now, history and discovery about the phytochrome. So first important work in this field was conducted in 1930s. A scientist name is L.H. Fleant and second one is E.D.M.C. Allister in 1935. They reported that seeds of lettuce lactua sativa did not germinate in dark, but they said that showed very good germination when exposed to water length from 5 to 5 to 700 nanometer in the red region. A scientist's name is H.A. Borthwick and S.B. Hendricks and other 1952 of the U.S. Department of Agriculture Plant Industry Station in Bells Valley, Maryland, using large spectrographs confirmed the findings of the Flint and M.C. Allister. Now talking on the distribution of phytochrome. Presence of phytochrome has been known in angiosperms, gymnosperms, liverworts, mosses, ferns, and some green algae. So, in which distribution of phytochrome in grass shoot is variable, but oat, rye, and barley seedling all have high concentration in apical regions of the coleopeptile or cellopeptile, you can say, near the shoot apex and in the growing leaf bases, in which phytochrome is present in most organs of all plants investigating, including roots. So many in green lower plants, phytochrome is permanently localized in conformation in or near the plasma membrane. Now, some characteristics of phytochrome, in which number one is that action spectrum of the light needed for these responses shows a peak in the red at about 660 nanometer. These responses can be reversed by NB application of far red light soon after the red treatment. Sensitive spectrometers can measure a decrease in absorbance at 730 nanometer when sensitive plant tissues are exposed to red light. The change in absorbance is caused by the conversion of photoreceptor from one structural form to another. The red absorbing form changes to the far red absorbing form when it absorbs red light and back again when it absorbs far red light. Now you will see, if you read many books, there are two different chemical structures present in it that are interconvertible. They form are named by the color of light that they absorb mechanically. These two different chemical structures, which are interconvertible, name are one is PR and second one is PR form. The phytochrome PR blue red light and BFR blue green lights. They are interconvertible. So the form PR absorbs at a peak of 670 nanometer. Is the synthesized in dark grown seedling. When PR absorbed red light, it converted to the PFR form. The PFR form absorbs at a peak of 730 in nanometer. The PFR is the active form that initiates biological response. When PFR is absorbed for red light, it converted to the PR form. So degradation habit of PFR is known to occur by a high process in which PFR is first attached to a small protein. This name is ubiquitin. These protein ubiquitin attached to PFR or to other protein targets them for degradation and carry out degradations ATP, and three enzymes are required. These enzymes, number one is protease. These recognize the protein. Hydrolase targeted the proteins. Release free ubiquitin. This picture you can see inactive form to active form, in which red light 60 nanometer coming to the PFR, and you can see in second line quick conversion are seen, and PFR levels of sun rises increases. Then what happened? It forward to, or it becomes night, in which darkness, slow reversion to the inactive sites, and the level night is increases. So these are the interconvertible. Now mechanisms. You will completely understand about the phytochromes, how they work. Phytochrome is believed to acting on following ways. These are the following ways which totally happen. How to? Number one is, according to Goldstone and co-workers, hydroxylation of flavonoid compound is affected by phytochromes. Mohr, however, suggests that radiation influences some basic metabolic change in the cell, responsible for many different effects, and influences on flavonoid hydroxylation pattern presumably represent only one such chain. So, works of KV Thiman and co-workers suggest that the told phytochrome's action involves the synthesis of nucleic acids and enzyme, which may be responsible for the physiological changes. Mohr and others have found evidence to suggest that phytochrome effects are mediated by activation of genes that code for specific messenger RNA molecules, which in their turn code for specific enzymes. There are certain responses due to red or far or red light which are too fast to be explained by the gene activation or repression hypothesis. Now the role of phytochrome. Many flowering plants use it to regulate the time of flowering based on the length of day and night. So you totally know that many plants are growing. By the reason is that they carry the carbon dioxide in the night and they provide oxygen in day. So this working. So length of day and night photoperiodisms. They call photoperiodisms and to set circadian rhythms in which circadian rhythms are working. So it also regulates other responses including the germination of seeds, elongation of seedlings, the size. So circadian rhythms, what they do, the germination of seeds and also the elongation of seed, so and also size. That's why plants growing shape and number of leaves, the synthesis of chlorophyll and the strengthening of the epicotyle or hypocotyle hook of dicot seedlings. When the light effect on the leaves, they convert the pigment into green with the help of the chlorophyll, which get by the sun and which is strengthening and giving effect force or power to epicotyle or hypocotyle hook of dicot siblings. Thank you so much.